Starfish. Majestic, beautiful, and voraciously carnivorous. Hunting down clams, oysters, mussels, small fish, snails, and barnacles, sea stars stop at absolutely nothing to get their next meal. Though they have a soft skin, spines of a sea star protect it from predators and also offer it structural support. The crown of thorns starfish, or Acanthaster plancey, on the other hand, eat a more sedentary meal, coral reefs. They often travel in packs, moving from reef structure to reef structure, depleting and destroying the beauty of all coral reefs along the way. The crown of thorns starfish is similar in basic biological makeup of other species of starfish, with radiating arms coming off of a central disc. Where crown of thorns starfish begin to differ from other starfish is in the number of arms. The average starfish typically has 5 arms, where the crown of thorns contains up to 21 arms surrounding the central disc. The typical 5-fold symmetry that is shown in starfish is only present during the pupal and early growth, until it starts adding more arms after the 5th is developed. The size of the starfish ranges from 9 to 14 inches, making them one of the largest species of starfish in the world. And the color of the organism tends to be a darker, sandier color, but depending on protection needs, they may exhibit brighter or more vibrant colors to scare away predators. The aboral side of the organism contains sharp spines, while the oral side contains blunt spines. The sharp spines on the aboral side are able to pierce through soft objects and are also able to release saponins that act as an irritant and also give off an unpleasant taste that make many predators avoid the starfish. It was previously thought that chemoreceptors were the crown of thorns starfish's main sense of finding food, but recently it has been discovered that on each arm, compound eyes allows for the organism to sense prey and reef structures. The paper that was focused on sought out to establish the role of Acanthaster plancey's eyesight versus their chemoreceptors in locating coral reefs. The researchers collected Acanthaster plancey from various areas around the island of Moria, French Polynesia, and deprived them of food for 2 to 4 weeks. A large lagoon, located 50 meters from the shore of Thame Beach on the island of Moria, was used as the site for the experiments in November and December of 2013. The site was 50 meters away from any coral reefs that could interfere with the experiments. Sea stars were put under anesthesia and the optic cushions, or their compound eyes, were cut off with scissors. The optic cushion is located at the tip of the sea star's arm, and it is the endmost tube foot which is modified to bear an eye. For each trial, a single specimen from each group were placed next to each other at 3 different starting points, 1.25 meters, 2.5 meters, or 10 meters from a live reef structure. The reef had a constant current running to ensure that the consistent chemical signals from the reefs reached the starting points. The pair were allowed to move freely until they either moved double their initial distance to the reef structure, or they reached the reef structure itself. The direction of each of the starfish's movement was recorded, and the mean direction of each trial group and starting distance was analyzed. The experiment found that the mean direction of movement at start points greater than 2.5 meters for the control group was always towards the reef structure, while the blinded starfish was downstream of the reef. When the pairs were within the distance of 1.25 meters, the blinded sea star began moving toward the reef. From a distance of 2.5 meters and 5 meters, the control group found the reef more often than the blinded, while in distances of 1.25 meters, both groups moved towards the reef. Starting in the 1960s, Acanthaster plancey's populations grew exponentially, and at one point over 13 million of these animals roamed the Indo-Pacific region. As the starfish eat more and more coral reefs, up to 10 meters squared or 30 feet squared per year, coral reefs are being consumed too quickly to regenerate, and algae takes over the coral skeleton. The population boom may have been caused by a few things. Humans overfishing more of Acanthaster plancey's predators like the Triton's trumpet and Pseudocoronactis, predators dying from habitat destruction, and also warmer sea temperatures due to global climate change, which leads to enhanced larval development. Proper removal of the starfish becomes a complicated issue, because not all methods are safe or successful past the initial removal of the organism. It originally was done by cutting up the sea stars and leaving them to die. This practice, while making them more vulnerable to predators, is also multiplying the numbers as they are able to regenerate themselves if enough of their DNA is. Another issue arises when trying to remove the starfish and take them out to kill them. While it may seem like the most plausible method, crown of thorns are very dangerous to handle. Contact with humans involves stinging pain that can last for several weeks, constant bleeding, and even surgery if the spine breaks off within the skin. One method that is very successful at controlling the organisms is injecting the starfish with a poison that can kill them. This has a very high success rate, but also becomes an issue as the poison is not kept within the dead sea star and will leak into the water, releasing heavy metals from the poison into the open water. On a positive outlook, a study was done in 1972 by James Porter that found although they are eating the coral reefs, they may have a positive impact on it as well. By continuing to eat the coral reef, it creates a more diverse area of coral. The study found that the crown of thorns starfish helps keep one species of coral from taking over the entire reef.